All of you, dear students, today's topic is morphology, anatomy and reproduction in Pinus. So far the introduction of the Pinus is concerned, it is the most important genus of the family Pinaceae and is represented by 90 to 100 species in the world. Its species usually grow on the slopes of hills and from dense and extensive forests of evergreen trees in the northern temperate regions of the world, sometimes extending up to the subarctic regions and in the tropical hills. Pinus is of great economic value. It is the main source of resins of various types and of various woods that we use as timber. Seeds of some species are a source of food. In India, the genus is represented by 6 naturally growing species. Pines are evergreen, resinous trees or really shrubs growing 3 to 80 meter tall, with the majority of species reaching 15 to 45 meter in height. The smallest pine are the Siberian dwarf pine, Potosi Pinyon, and the tallest is 81.79 meter tall, Ponderosa pine, located in the southern Oregon. Pines are long-lived, typically reaching age of 100 to 1000 years, some even more. The bark of most of pines is thick and scaly, but some species have thin, flanking bark. The branches are produced in irregular pseudowhorls, actually a very tight spiral, but appearing like a ring of branches arising from the same point. Many pines are uninodal, producing just one such whorl of branches each year from buds at the tips of year's new shoots, but others are multinodal, producing 2 or more whorls of branches per year. Shoots are dimorphic, with long shoots and dwarf shoots. Dwarf shoots are borne in close spirals from axils of scaly bracts and bear fascicles of leaves, needles. Branchlets are stout, ending in a compound bud with many bud scales. Leaves are dimorphic, spirally arranged. Foliage leaves are in fascicles of 1 to 6 depending upon the species, 2 to 3 angled and are rounded on abaxial surface. The foliage leaves may persist from 2 to 12 or more years. They are sheathed at the base by 12 to 15 overlapping scale leaves. Anatomically, the stem shows a considerable advance over the cycadales, like the stem of dicotyledons. It has a concentric arrangement of primary permanent tissue. It is composed of a small pith, a thick vascular slender made of a ring of separate collateral and open vascular bundles, a thin cortex and an epidermis. Pines are mostly monoecious, having male and female cones on the same tree, though a few species are subdioecious. The male cones are small, typically 1 to 5 centimeter long and only present for a short period, falling as soon as they shed their pollen. The female cones take 1.5 to 3 years depending on species to mature after pollination, with actual fertilization delayed by 1 year. At maturity, the female cones are 3 to 60 centimeter long. Each cone has numerous spirally arranged scales, with 2 seeds on each fertile scale. The scales at the base and the tip of the cone are small and sterile, without seeds. The seeds are mostly small and winged and are anemophilous, that is wind dispersed, but some are larger with only a vestigial wing and are bird dispersed. At maturity, the female cones usually open to release the seeds, but in some of the bird dispersed species, for example, a white bark pine, the seeds are only released by the birds, breaking the cones open. In others, the seeds are stored in closed cones for many years until an environmental cue triggers the cones to open, releasing the seeds. Now, we will see the distribution of Pinus. Pines are native to most of the northern hemisphere. In Eurasia, they range from the Canary Islands, Iberian Peninsula and Scotland up to the Russian forest, and in the Philippines, Finland, Sweden, the Himalayas and southern Asia. In India, it is represented by 6 naturally growing species. As per the morphology of the pines is concerned, pines tree are varying in height from 20 to 65 meter and reaching a diameter of about 4 meter or even more. Pinus ristata from California is about 4,000 years old. It grows on the slopes where rainwater is easily drained off. As in the pteridophytes, the pinus tree is a sporophyte. It is differentiated into roots, stem and leaves, which are described as: first, root. The primary root persists and forms a typical elongated straight taproot. In case the plant is growing on the shallow soil overlying rocks, which is impossible for the taproot to penetrate, it remains short and develops strong lateral roots, forming a massive root system spread out over an extensive area. Tip on the root has a protective root cap. As the root system becomes fully developed, the new young branches remain short and undergo frequent forking, so that clusters of small rootlets are formed. Some of these branch dichotomously, form coralloid mosses with ectotropic mycorrhizae. These are the mycorrhizal roots. Under the microscope, they are seen to be covered with a close weft of colorless hyphae. Some of these penetrate the outer tissue of the young roots. The exact relationship between the hyphae and the root system is a debatable question. Some physiologists consider the fungus to be merely a parasite. Others hold that both the partners are mutually beneficial to each other. The fungus receives food from the host root. In turn, it increases the water absorption by roots and thus takes up the function of the root hairs and makes available to the host some of the nitrogen in the form of organic compounds of the soil. The mycorrhizic roots differ from the normal roots in the following respects: they are short and thick, they lack root hairs, they lack a root cap, they are more extensively branched or occasionally unbranched, they are covered with fungal hyphae. Now, we will see the morphology of stem. Typically, the stem displays the exicurrent habit. It is tall and straight and covered with a rugged scaly bark. The main stem is branched. The branches are of two types: a long branch and short or dwarf branch. Long branch. These are the ordinary or normal branches. They grow indefinitely in active growth by means of an apical bud. They are often called the shoots of unlimited growth. They are covered with brown bud scales. These branches arise as buds in the axils of scale leaves at the end of each year's extension growth. In the following year, these buds develop as an apparent whorl of branches. Short branches. In addition to the main branches or long branches which arise directly from the trunk, there are very numerous branches of limited growth borne on the ordinary branches in the axils of scale leaves. They are often called the dwarf shoots or shoots of limited growth or suppers. Each dwarf shoot consists of a short axis terminating in a cluster of 1 to 6 green needles or foliage leaves. Now, leaves. The leaves are also of two kinds. The first kind of leaves are somewhat of unusual type, being long, narrow, acircular, tough and green, and are frequently known as pine needles. Their surface is smooth. They are not borne on ordinary branches, but appear only on dwarf shoots in clusters of 2 Pinus mercusi, 3 Pinus roxburghii, or 5 Pinus wallichiana, according to the species. In Pinus monophylla, only one leaf is produced on the dwarf shoot. The dwarf shoot bearing the foliage leaves are known as suppers. A thin, dry, membranous sheath surrounds the base of each supper of the needles. The needles are persistent They fall only when the supper is shed as a whole. Hence, the pine tree is evergreen. Secondly, there are the scale leaves. They are brown, membranous, and are protective in function. The scale leaves are only ones borne on the long branch. They are found on dwarf shoots as well. They fall off as the branches mature. The reduced size and sclerotic character of the leaves have been associated with the xerophytic habitat of the species. First, we will see the primary structure of the stem. Anatomically, the stem shows a considerable advance over that of Sacadales. Like the stem of dicotyledon, it has a concentric arrangement of primary permanent tissue. It is composed of a small pith, a thick vascular cylinder made of a ring of separate collateral and open vascular bundles, a thin cortex, and an epidermis. It covers the outer surface of the stem and is single layered and irregular in outline. The epidermal cells are heavily thickened with walls strongly chitinized. Cortex. The cortical cells beneath the epidermis are strongly thickened to form the hypodermis. The inner cortex consists of thin-walled parenchymal cells. It is traversed by large region ducts or canals which run both radially and longitudinally through the stem. Each duct or canal is lined by a layer of thin-walled parenchymatous, glandular, secretory cells constituting the epithelium. Neither endodermis nor pericycle is recognizable. Primary vascular tissue. It consists of a ring of 5 to 6 separate primary vascular bundles. The bundles are collateral and open. They are like closed together so that the primary medullary rays connecting the pith with the cortex are narrow and parenchymatous in nature. Each bundle has a primary phloem on the outside and primary xylem on the inner side, and primary cambium in between the two. The primary xylem contains neither true vessels nor wood fibers, which are characteristics of angiosperms. It consists of tracheids, which are arranged in uniform radial rows, and xylem parenchyma only. The phloem contains sieve tubes and phloem parenchyma cells. Companion cells are absent. Albuminous cells also occur and have the same relation to the sieve cells as of the companion cells. The primary cambium, which is located between the xylem and the phloem of each vascular bundle, consists of a single layer of meristematic cells. It provides continuous growth in diameter. Pith. Within the ring of the vascular bundles is the parenchymatous pith. It forms the core of upright woody stem of Pinus. Now, dear student, we will see the secondary structure of Pinus and its anatomy. The secondary tissue are formed in the same way as in the dicotyledons. The primary cambium of the stele extends tangentially. The edges of the bundle cambia come closer and eventually come in contact. In this way, a complete ring of cambium is formed. The cambial cells divide repeatedly in a tangential direction. Two cells are formed at each division. Of these, one remains cambial. The other, which is called the tissue mother cell, may divide once or twice. The cells formed become differentiated into xylem or phloem according to their position on the inside or outside of the cambial cells. Such growth is known as secondary growth. The new xylem and phloem cells thus formed constitute the secondary xylem and secondary phloem respectively. The cambial cells in the meantime increase in size and again divides. As a consequence, a continuous ring of secondary xylem on the inner side and a layer of secondary phloem on the outer side is formed. The cambium remains as a permanent meristematic between them. Periderm or cork. The formation of secondary vascular tissue stretches the outermost tissues of the xylem or outermost tissues of the stem, which eventually ruptures and are peeled off as thin strips of scaly bark. Before this takes place, a special protective tissue, the periderm or cork, is formed. It is formed by a layer of cortical cells near the outside. These cells become meristematic and constitute the cork cambium or phylogeny. It is secondary merist. The cells of the cork cambium divided by two parallel walls. The inner cells form the additional cork, whereas the outer constitute the periderm or cork. The latter tissue serves to protect the delicate tissue lying within it when the epidermis is peeled off. It is impervious to water and thus helps to prevent excessive evaporation. It may be mentioned that cambial activity, xylem and phloem differentiation and formation of cork cambium are all hormone controlled. Now, anatomy of the root. It consists of piliferous layer, cortex and stele. The arrangement of tissue is in general similar to those of roots of dicotyledons. Now, piliferous layer. It is the outermost layer. The cells of this layer fit tightly against their neighbors. Now, cortex. Within the piliferous layer is the cortex. It is a wide zone of parenchymal cells and thus forms the bulk of the roots. It is bounded on the outside by endoderms. Now, endoderms. It is a single layered. The cells are of brownish orange color. They are suberized and are usually impregnated with tannins. Pericycle. It delimits the stele and is many layered. The pericycle cells contain starch and are usually impregnated with tannins. Now, we will see stele. It is diarch, occasionally triarch or tetrarch. The higher number is up to 6. An equal number of phloem bundles alternate with the xylem bundles. The protoxylem groups lie at the end of metaxylem, which occupies the center of the root. As the development of the metaxylem is centripetal in young roots, the central cells may be found unlignified, constituting the pith. Each protoxylem group is forked, so the xylem bundle appear Y-shaped. A resin passage lies between the arrows of the Y. A cross section through the mycorrhizal roots show that hyphae run mainly between the cortical cells. They lie thickly in the intercellular spaces. No fungus is present within the endoderms. Most of the hyphae, forming a complete layer over the surface of the root, are cut transversely. They present the appearance of the outer pseudoparenchymatous tissue. Now, anatomy of leaf. The leaf shows a complex and unusual structure very much different from that of the leaves of cryptogams as well as that of typical leaf of the angiosperms. Its anatomy suggests that it is adapted to endure severe environmental conditions. It is not flat but is about as thick as broad. In a cross section, it appears somewhat triangular. It reveals the following structure from outside to inwards. First, epidermis. Forming the outer boundary of the leaf is a single layered epidermis. The epidermal cells are thick walled and heavily cutinized, so that the epidermis is covered on the outside by a thick cuticle. The deeply sunken stomata are present on all sides of the leaf. Their guard cells are situated well below the level of epidermis, so that there is sunken pit leading to stomatal aperture. Hypodermis. It lies beneath the epidermis and is composed of one or more layers of thick-walled sclerenchymatous cells. The hypodermis is frequently interrupted by air spaces beneath the tissue. It is strongly developed at the angles. Mesophyll. Within the hypodermis is the parenchymatous mesophyll. It is compact and shows no differentiation into palisade and spongy tissues. It consists of thin-walled cells which contain numerous chloroplasts and abundant starch. The mesophyll thus functions as the chlorophyll-bearing tissue, manufactures food for the plants. Endoderms. The central tissue of the leaf is enclosed by a conspicuous single-layered endodermis, which delimits the mesophyll. The endodermal cells are large and oval and possess Casparin strips. Pericycle. Within the endoderms is the many-layered pericycle. It consists of many types of cells. Vascular bundles. Embedded in the pericycle are two vascular bundles. They are separated by a band of sclerenchymatous fibers. The two bundles run closely parallel from the base to the apex of the leaf and are unbranched. In some species, there is only a single bundle. Each bundle contains xylem and phloem. The phloem faces the curved lower or outer side. The xylem above the phloem facing inwards towards the pointed end. The bundle is thus collateral. Now we will see its reproduction. Vegetable reproduction is unknown in pines. They reproduce by means of seeds. Reproduction by seeds in wallow, reproduction by both spores and gametes. It also comprises the origin, growth and development of the gametophytic generation. Now reproduction by spores. All seed plants are heterospores, which means that the reproductive organs called spores are of two kinds: microspores, that is pollen grains, and megaspores, that means embryo sacs. They are produced in specialized structures called microsporangia, pollen sacs, and megasporangia, ovules minus their coat respectively. The sporangia are born in sporophylls, which are organized to form cones or strobili. The latter are of two kinds, one containing only microsporangia, the other megasporangia. It is customary to call the former as male cones or microsporangiate strobili, and later female cone or megasporangiate strobili. It is more appropriate to call the one as the microsporangiate or staminate and the one as megasporangiate or carpellate cone or ovulate cone. Pinus is monoecious. It bears both types of cones on the same tree on separate branches. The microsporangiate or staminate are male cones. They occur laterally in clusters, each in the axil of scale leaf at the base of a terminal vegetative bud which they surround. They are concealed amidst last year's foliage. They can be seen on the pine tree in the beginning or middle of March in the hills, and in January or February in the plains. The male cone is strongly, shortly stalked and consists of elongated central axis bearing a number of spirally arranged and closely fitted scale-like microsporophylls. The latter are equivalent to stems of angiosperms. Each microsporophyll takes its origin from the central axis, runs out horizontally to end in a sterile flattened head. It bears two microsporangia or pollen sacs as swellings on the lower surface of its horizontal position. The microsporophyll is attached to the axis by a short stalk. The microsporangia are filled with numerous winged microspores. A number of scales at the base of the male cone remain small and sterile, usually 8 to 10. The young cones are green or reddish purple, but becomes yellow or reddish brown at the time of shedding. Megasporangiate or ovulate cones. They are fewer in number and arise single or in a small cluster of 2 to 4. When young, it is small, erect, reddish to pinkish structure, which stands erect on a short stalk covered with scales. Like the male cone, it consists of a central axis which bears paired scales in a closed spiral. The lower scale of the pair is small, called the Bract scale. It is leathery and directly attached to the cone axis. The upper scale of the pair is larger, thicker and stouter. It is ovuliferous scale. It develops from the upper surface of the Bract scale and bears two ovules side by side. On the lower side as two small white swellings. There are about 80 to 90 megasporophylls in a single female cone. Now we will see the pollination in pines. It is affected by the wind and is therefore anemophilous. It takes place towards the end of May or during the month of June in the hills. In plains or at lower altitudes, it takes place towards the end of February or mid-March. At this time, the ovulate cone is young. It is about the time of meiosis, which means that megaspores have been established. The development of the embryo sac is subsequent to this event. The ovuliferous scales, which prior to pollination fit tightly over one another, opens out, providing a ready access for pollen grains to the ovules. The microsporangium or pollen sac opens by a median longitudinal slit along its underside. On a slight shaking, the tree which is producing pollen, will emit clouds of yellow dust which arise in the air and color the whole landscape in the pine forests. The yellow dust is formed by the countless number of pollen grains. They are light in weight and buoyant due to the wings. Upon liberation, they float upon the air and settle down only slowly. Much of the pollen is wasted. Those that chance to be blown by the wind to the female cone are trapped between the scales. Here they are caught in a drop of sticky substance, the pollination drop exuded from the micropylar end. After pollination, the oleferous scale closes up again and the stalk of the cone curves so that it becomes inverted. Now we will see about fertilization. The pollen tube, whose growth is arrested in the winter of the first year, after it has penetrated a short distance into the nucleus, resumes growth again in the month of April when the female cone is running into its second year. The generated cell divides into a stalk cell and a body cell. The body cell then divides into two male gametes or microgametes, but no wall is formed separating the cytoplasm of the pollen tube. The male cells are non-motile. In this respect, these differ from the motile gametes of the cycads. They float within the pollen tube. Meanwhile, the pollen tube has passed entirely through the nucleus and reaches the female prothallus. Finally, the tip of the pollen tube enters the neck of the archegonia. The two non-motile cells and the other contents of the tube are discharged into the venter, where all the discharged contents and the cells disintegrate except one male cell. The nucleus of the surviving male cell unites with that of the egg to accomplish fertilization. This process is usually completed in the last week of June. The fertilized egg secretes a wall around it and becomes the oospore or zygote. The zygote therefore is the first cell of sporophyte. More than one archegonia may be fertilized in a single ovule, but only one comes to maturity. Others perish. The zygote immediately germinates. It undergoes a series of divisions still enclosed within the ovule to form a relatively elaborated proembryo. 4 potential embryos are formed from one fertilized egg. In the course of further development of the potential embryos, one takes the lead and grows more rapidly than the others. The actively growing potential embryos, by cell division, cell growth and differentiation, becomes the embryo of the seed. Others die off. Now we will see the alteration of generation. In the cycle of Pinus, the sporophytic phase is of long duration and is represented by the huge pine tree which bears the staminate and ovulate cones. The staminate cone produces pollen grains in pollen sacs on the microsporophylls. The female cone producing megaspores, one in each ovule, borne on the upper surface of the oleferous scale. Since meiotic division occur at the time of the differentiation of the pollen grains and megaspores, the later structures represent the first cell of the second generation, the gametophytes. Both the gametophytes, male and female, in Pinus are parasitic upon the sporophyte. The little microgametophyte is carried by the wind to a position in the vicinity of the ovules, from where it is shifted to the apex of the nucleus. It then puts out a pollen tube which carries the male gametes, to which the archegonia develops at the micropylar end of the female prothallus. The male gametes unite with the egg in the archegonia to effect fertilization. The fertilized egg is a diploid structure and is the first cell of the sporophyte. It develops into the embryo, which is the new sporophyte. With this, I think we have completed all the aspects of the Pinus. Its morphology, anatomy and reproduction all have been completed. With these words, I would like